Hello students, welcome back to the session. In today's session, we'll discuss the regulation of gene expressions. So you already know that. What is gene? A gene is a fragment of DNA, that carry genetic information from one generation to the another generation. And that gene is expressed in the form of polypeptide or proteins. For example, in case of pea plant, the tall character is controlled by capital T. If that character appears in the next generation, that particular gene is expressed, and that particular gene is expressed in terms of proteins or polypeptide. And the regulation in case of a living organism differs from prokaryotes to eukaryotes. In case of eukaryotes, the regulation of genes occurs in 4 levels. One is a transcriptional level. At the time of formation of mRNA or heterogeneous RNA, or we call it as a primary transcript. One. Second one, splicing level. You already know sequences. One is coding sequences and the other one is non-coding sequences. The coding sequences are called exons, and non-coding sequences are called introns. So during protein synthesis, only exons are required, introns are not required. At the time of protein synthesis or at the time of splicing, the regulation of gene expression is controlled. Next one. During the export or transport of mRNA from nucleus to the cytoplasm. Third level, and fourth one, translation level. So these are the 4 levels of regulation, gene regulation. The next question arises: if all genes were not regulated, what happens? So if all genes are not regulated, all genes in a DNA will be expressed. If all genes are expressed, they will produce many proteins. Large number of proteins are produced. For example, we have 30,000 genes. If all genes are expressed at the same time, 30,000 different types of proteins are produced. Number of proteins are produced at a time, single at a time. So this is one. So cell would require a lot of energy. Whenever it is required, that particular gene is expressed. If it is not required, that particular gene is suppressed. Why? Because both transcription and translation, they require large amount of energy. That's why the gene is expressed at a specific time when it is required. Then only that gene is expressed. So third one. And fourth one, this as a result of if all genes are expressed, that would make all the cells an eukaryotic identical. So these are the 4 reasons. That's why all genes are not regulated at the same time, or not regulated at the same time. Let us start with the first one. the Operon concept. Operon concept means what? Operon means a set of genes which regulates the metabolic pathway. We call it as the operon. There are two types of operon. One is inducible operon, another one is suppressible operon. So in the presence of inducers, that particular gene is expressed. In the absence of inducer, that particular gene is suppressed. No need of expression. If inducer is present, that particular gene expressed and produce specific proteins. If inducer is absent, no need of that gene to express itself. For example, lactose, or we call it as a maltose. We will take maltose. It's a disaccharide. In order to digest maltose, we required an enzyme called maltase enzyme. So this maltase enzyme is nothing but a protein. The gene which is responsible for production of this enzyme is expressed when maltose is present. In the absence of maltose, that gene is not expressed, that is suppressed. Is that clear? So when maltose is present, that gene is expressed and that produces maltase enzyme, and maltase enzyme which act on maltose and converts into 2 glucose molecules. Got my point? So maltose is an inducer. So set of genes are activated when inducers are present. So inducible operon and suppressible operon. Look at here. The Operon concept was first explained by Francis Jacob and Jacques Monod, or Jacob and Monod explained the Operon concept in the year 1961. So all set of genes controlling one metabolic pathway constitute operons. When you look at the primary transcript, you already know that the transcriptional unit has promoter, structural genes and terminator. The structural genes can be regulated by a set of genes, and those set of genes are called regulator genes. So structural genes along with the regulator genes together to form operon, and that operon controls one metabolic pathway. So the genes in a cell are expressed to perform specific function or set of functions. I took only one example. If maltose is present, in order to digest maltose, what we required? We required maltase enzyme. So in the presence of maltose, that particular gene is expressed and that will produce mRNA first, and that mRNA will produce synthesize enzyme called maltase, and this maltase enzyme will act on maltose and converts into 2 glucose molecules, glucose plus glucose. Next one, for example, one more example. The cell produces required protein at a required time, and it will produce the required amount. Okay, so all the time it will not be expressed, it is suppressed. Okay, in the presence of inducer, that particular gene is expressed. So E. coli is cultured in a medium containing glucose, and this glucose is utilized, is a source of energy, and use glucose as a source of energy and carbon. Immediately it will multiply rapidly. When E. coli cells are transformed into the other media. So in the presence of glucose, cells multiply rapidly. When such culture, okay, if bacteria are transferred into the other media containing lactose media, it will take some time. The question arises, why? Because cells multiply only after a short interval of time, because it prepares itself. What happens? A new medium requires synthesis of new enzyme. That means, so in the presence of lactose, glucose is required for multiplication. So in the presence of lactose, multiplication stops temporarily. So what cells or genes will do? The genes will produce a lactase enzyme, and that lactase enzyme which act on lactose and converts into glucose plus galactose. So cells will synthesize enzyme called, okay, there are three different types of enzymes are produced. One is a lactose permease enzyme, okay, which helps in transport of lactose from medium into the cells. Second one is a beta galactosidase enzyme. This hydrolyzes lactose into glucose plus galactose. And third one is trans-acetylase enzyme, which detoxify compounds. So these are the set of enzymes synthesized. Soon after synthesis of these enzymes, okay, what happens? Immediately multiplication starts. Okay. Look at here. When the bacterial cells are transferred into the glucose media, immediately they start multiplying. Same bacterial cells are transferred into the lactose media. It will take time because it will synthesize set of enzyme, and those enzyme, okay, which converts lactose into glucose plus galactose, and that glucose is utilized for multiplication. That means, so in the presence of inducer, the particular genes are expressed. So next, so presence of lactose induce enzyme formation. If lactose is absent, no need of production of these enzymes: permease enzyme, beta galactosidase enzyme, and trans-acetylase enzyme. So presence of lactose induce enzyme formation. That's why it is considered as a inducing substance. So lactose is called as an inducer. So in the presence of inducer, these three set of enzymes are produced, and as a result of this, lactose metabolism takes place. Look at here. Lactose is a substrate for the enzyme beta galactosidase, and it regulates switch on and switch off of the operon. So in the presence of lactose, switch on mechanisms takes place. In the absence of lactose, switch off mechanism. That means the set of genes are not expressed in the absence of inducer. So hence it is termed as lactose, termed as the inducer. Absence of lactose, operon regulates enzyme formation. No need of enzymes in the absence of lactose, or in the absence of inducer, no need of enzyme production. The operon consists of a set of genes. One is structural genes. Okay. So structural genes, in case of prokaryotes, it's already known that the structural genes are polycistronic. That means the structural genes, so structural genes are polycistronic. That means they have ability to synthesize many proteins or polypeptides. Whereas in case of eukaryotes, the structural genes are monocystonic. Okay, they have ability to synthesize only one polypeptide chain at a time. So these structural genes are regulated by two genes, promoter and regulator genes. Promoter and regulator genes. So three genes are close together, structural genes. One is LAC-Z, LAC-Y and LAC-A. So single polycystonic multigenic mRNA with the initiator, terminator and coding sequences. LAC-Z, which codes for beta-galactosidase enzyme, which hydrolyze lactose into glucose plus galactose, and LAC-Y, which codes for permease enzyme, which increase the permeability of lactose from medium into the cells, and LAC-A, which codes for beta-galactoside transacetylase, which detoxify the compounds. Second one is promoter. So we already discussed in the process of transcription. A promoter is a site where DNA dependent RNA polymerase enzyme binds and initiates a transcription process. So it lies close to the operator genes. So what is the significance of operator gene? Look at here. These operator gene located very close to the promoter region, and just besides the operator gene we have a three structural gene, LAC-Z, LAC-Y and LAC-Z, Y and A gene. And this operator gene which regulates these structural genes. So promoter is a site where RNA polymerase enzyme binds and initiates a transcription process. And it is made up of 6 bases, which is commonly known as Tata box or Pribnow box in therapy. It is named after the discoverer. One, structural gene. Second one is gene. Next one, regulator gene, or we call it as the inhibitor gene. In case of prokaryotes, the regulation of gene, or regulation of gene expression is controlled by a specialized protein that is called accessory protein. And that accessory protein, or we call it as a repressor protein, produced by regulator gene, that is the inhibitor gene. What happens? The repressor gene undergo transcription and will synthesize mRNA, and that mRNA is translated and synthesize repressor protein, and the binding site of repressor protein and operator gene are complementary. So this regulator gene, that is inhibitor gene, will produce a repressor protein, and the binding site of the repressor protein is complementary to the operator gene. When this repressor protein go and bind to the operator gene, these structural genes are not expressed, they are suppressed. Next one. It controls transcription of LAC-Z, Y, A gene. So repressor has complementary sites. So it go and binds to the operator 1, and the repressor protein has a complementary site for inducer also. It go and bind to the inducer, that is lactose. Is that clear? So this is about the structural genes and Operon set of genes. Next one, we'll move to there are 2 mechanisms, switched on and switch off. So in the presence of inducer and in the absence of inducer. Let us start with the first one. In absence of inducer, what happens? So in the absence of inducer, that is lactose, what happens? So this regulatory gene or inhibitory gene will produce a repressor protein, repressor protein, and this repressor protein, in the absence of inducer, go and bind to the operator genes. So repressor protein binds to the operator site and prevents transcription of structural genes. As a result of this, these structural genes, okay, will not synthesize proteins or enzymes. I told you, okay, these structural genes synthesize set of enzyme. One, Z codes for beta galactosidase, Y, okay, permease, A, okay, trans acetylase. No synthesis of proteins, okay, proteins takes place. So lac operon, okay, is switched off. Next one, it is repressed. The structural genes are suppressed or repressed in the absence of inducer. So in the presence of lactose, what happens? In the presence of lactose, inhibitor genes will produce repressor protein, and that repressor protein go and bind to the inducer, that is lactose. So when inducer go and bind to the repressor protein, this repressor protein unable to bind the operator gene. So binding causes physical changes in the repressor. So repressor is an inducer complex unable to bind the operator site. Unable to bind the operator site. Look at here. In the absence of inducer, this repressor protein go and bind to the operator gene. So as a result of this, these genes are, okay, they are switched off. In the presence of in the presence of lactose, what happens? The repressor protein go and bind to the inducer, and automatically what happens? This repressor protein unable to bind the operator's site. As a result of this, the structural genes get activated. So RNA polymerase enzyme binds to the promoter site, and LAC-ZYA gene transcribed into mRNA, and that mRNA will synthesize 3 set of enzymes: lactose permease, beta galactosidase, trans acetylase. And when these enzymes are synthesized, automatically what happens? Lactose is converted into glucose plus galactose. This will be called as a switched on. So you remember one thing, whenever inducer is present, that particular gene is expressed in terms of protein, we call it as a switched on. In the absence of inducer, that particular gene is repressed with the help of accessory proteins, or we call it as a repressor protein, that we call it as a switched off. So this completes the regulation of gene expression. In the next session, we'll discuss human genome project. Thank you.